keeps on coming back. Now you see me, now you don't. First you leave and then you won't disappear. Saw you at the store till you turned around. Then it wasn't you anymore, but I found and refound. It's a call for a walk or a call. First it's you, and then it's a go. Keeps on coming back. Now you see me, now you don't. First you leave and then you won't disappear. Oh, the other night I almost got to sleep without anyone who's not there visiting me. You can see me if you want, I left the light on. You can't miss it unless it's on. Let it go on. Keeps on coming back. Now you see me, now you don't. First you leave and then you won't disappear. Now you see me, now you don't. First you leave and then you won't disappear.
Hello everyone! Welcome to our stream today. I haven't streamed in a while. This is awesome. I'm super happy and excited for what we're doing today. My name's Ilea Mariam Pig, better known if you're at this channel, you probably already know, Green-Eyed Trombonist. And I, as some of you might not know, am a grad student at University of Wisconsin-Madison working towards a PhD in communications. And I was supposed to do a few conferences this last spring. I can think of 3 off the top of my head that I was going to be doing things at, presenting research. But then COVID happened. And the university made the correct decision to close campus, have people go home, try and reduce the chances of infection. But another side effect that went into effect because of COVID is that conferences, academic conferences, were canceled, basically. Very few of them attempted to do online conferences instead. Most of them canceled outright. And as a result, we have all of these grad students with super awesome research that wasn't able to be put out there into the community. So I thought that I would reach out to some grad students and um, interview them basically on my Twitch channel about their research, their interests, what they are passionate about what they're doing. Not only so that they get more presentation experience, not being able to do the conferences, but also so that people who don't attend academic conferences or who aren't able to attend those conferences can also become aware and share in those passions with them. I see Simon's in the chat. Good morning, Simon. If anyone else is in the chat, say hello. One thing that we are going to do is you will have the opportunity to ask questions of the people that I research. And I'm super excited for this first one that I'm talking to. Her name is Alexa DiNicola. She is a botanist at University of Wisconsin-Madison. And I'm not a very planty person, I will say. My dad has the green thumb. I say that I have a black thumb. He didn't pass that lovely gardening trait on to me. But I love people's passions. So even if you are like me and you know nothing about botany or how to keep even a cactus alive, hopefully you will listen to what Alexa has to say and get some really good information. Because I know I've had conversations with Alexa in the past, and it always blows me away how passionate she is about plants. So with that brief introduction thing, how about we introduce you all to Alexa? And Alexa, why don't you introduce yourself? You're muted, Alexa.
Yep, let's fix
There we
that.
go.
Hello, everyone. Sorry about that. Well, you know my name. You know what I study and where. I'm a grad student at the University of Wisconsin-Madison. And long story short, I study plant evolution. The deeply weird and wonderful things that plants do that develop their diversity and the sheer range of living variation. A lot of people don't think of plants as much more than scenery. Quite frankly, that's because we're too damn impatient to notice the things that they do. Most of plant activity is on slow time scales or very small spatial scales. And it's pretty wonderful once you get down to it. Specifically, I study cinquefoils, a particular group of little high mountain meadow plants. I've got a few with me today, that casually thumb their collective nose at the concept of species boundaries. They make sense in some very interesting ways.
That is awesome. I love the display of these plants that you have behind you. What did you call them?
They're called cinquefoils, the Brewer's cinquefoil complex. It's French for five-leaved. The flower on this one's a little wilted. Oh, there's a better one. If you take a close look at the flower, let's see if I can get my computer to focus on it, you can see 5 petals. That's extremely diagnostic for these plants. There's one species that has 4, but they're even weird among cinquefoils, which is saying something.
So you've explained a little bit about like introducing yourself and these plants. Why don't you briefly explain the research that you are specifically doing?
So the way I like to explain this is with a, to begin with a little analogy. One night, 5-7 years back, I came home from work tired and went, you know what, I want banana pancakes for dinner. So I made them. And I'm standing over the skillet about to flip this banana pancake, and I realized something really wonderful. These pancakes are mostly wheat and bananas, yeah? Well, most farmed banana plants are clones of each other, which makes them insanely disease prone. The entire world banana industry has been wiped out by a single disease before, and it's going to happen again.
I would like to insert, this is what happened with the potato famine.
Mm-hmm, same deal. If you've ever heard the song, Yes, We Have No Bananas, that was exactly why. And then you got wheat, which is the product of 2 hybridization events that shouldn't have worked. And as a result, to make a very long story short, is literally working off the complete fused sets of genes from 3 separate species. So I'm standing here over this skillet of doomed clones and frankengrains and realizing that relative to my daily life, the only weird thing about this meal is that it's breakfast food for dinner. That should give you some idea of just how gorgeously bizarre plants are. There's a story like that behind every sidewalk weed and half the food in your kitchen. What I study is the forces that create and shape all this plant biodiversity. They are huge and intricate and really not well understood, especially when it comes to how they affect not just the plants themselves, but also each other. So that's where my thesis work comes in. I study the Potentilla brewer complex, brewer cinquefoils, a group of 5 odd, well, depending on who you ask, it's anywhere between 5 and like 15 species of these high mountain meadow wildflowers, vaguely strawberry-like. And in them, they're a great case study for this stuff because in them, several of these big forces are all being weird at once. For example, they hybridize. I mean, they hybridize a lot. There is pretty good field evidence that they trade chromosomes like baseball cards. They should all be blending into one big species. That's what happens when things hybridize freely like that. But they stay quite distinct. More than one spot in the field I can point to, you'll find a pretty consistent population of one species, a pretty consistent population of another species, and the whole smear of hybrids between them, which is not something that should work long term. How are they keeping that going? Good freaking question. Very much something I'm working on answering. The shapes of them, the leaf shapes, are another interesting challenge. They have leaves that are pinnate, as in you have a single stem with individual leaflets coming off the sides. They have leaves that are palmate, where all the leaflets come from a single point, like a marijuana leaf. And then they actually have leaves that are kind of in between. I have even seen pinnate and palmate leaves on the same plant, which isn't really a thing that happens, except in these guys, because that makes perfect sense. I'm actually having to develop some new math methods just to measure and compare them very well, much less figure out how they grow that way or why. And then there's where they live. These guys are mountain species. They are not well adapted to xeric climates. They all insist on having a lot of water for at least part of the year. But most of the area where they grow is, Eileen, could you pull up that range map?
Sure.
Most of that area is high desert. So they grow on these isolated, scattered peaks. It's a chain of sky islands embedded in a sagebrush sea. I figure at the end of the last ice age, they'd all have been one widespread lowland species. And as the glaciers melted, the desert formed, they'd have been pushed gradually up the mountainsides as the valleys got hotter and hotter, where they would have eventually diversified. So having any one of these oddities would make a study group deeply interesting. But the really good bit is in having them all at once. It means I can study not just how plants evolve, say, in sky islands, or when they hybridize all the time, but how the sky island habitat affects hybridization and vice versa. Do the mountains restrict what other plants are there to cross with? Does hybridization stir up the gene pool so they can better adapt to new mountaintops or changing mountaintops? By studying each force in light of all the others, my thesis will help us make sense of them as a whole and therefore the diversity they create. It's going to be awesome, provided my study subjects last long enough. At the end of the last ice age, they climbed mountains once to escape a round of global warming. But this time, A, it's happening a couple orders of magnitude faster, and B, there's not a lot of mountain left to climb. They're not alone either. Show me a peak in the Rockies and I'll show you 10 species that will be dead by 2100 at our current rate. My hope is that by figuring out just how these guys survived and thrived through the end of the last ice age, I can help figure out how to get them and others like them through the next big round of change. That way, to bring it back, when our grandchildren take their seats at the global table, there might just be some banana pancakes left for them too.
I think anyone who loves bananas would love that.
It's a useful analogy. Banana pancakes has kind of become my personal shorthand for the kind of wonder that smacks you in the face completely unexpectedly.
Mm-hmm. So, as I've said, basically, botany is completely foreign to me, right? Um, there's a lot of things that I'm interested in, and I am interested in plants, but from like a I'll be over here not killing them kind of perspective. Um, but what was it that got you into botany?
Well, worth pointing out, botany and horticulture are very different things. The only reason I can keep these so many plants alive for so long and so well is because I haven't, the botany department has an excellent greenhouse staff.
Mm.
I have been known to kill cacti too. That said, I have literally always loved plants, as in I have evidence of being nuts about my backyard flora from before I have actual memories. Um, Little poems, drawings, uh, get written down by my parents, of course. I was like 3. Um, but how I got into it academically, well, I was a philosophy major at the time, my 3rd year of college, and spring came late. At the same time, I had a metaphysics and a philosophy of science class. So spring comes late, and I'm sitting in the corner basically jonesing. Why is it not green yet? Why is it not green yet? Where is spring? Come on, guys. That's 1. 2, in the philosophy of science class, we had just spent a good couple days proving through a pretty unassailable chain of symbolic logic that if you hold a position and are shown believable evidence that contradicts that position, you are fundamentally and demonstrably wrong not to shift your position towards that evidence. And yet at the same time, in the metaphysics class, we had just spent a week discussing the mind-brain correspondence, and everybody looked at me like I'd just sprouted ear turnips when I suggested that neurology might have something useful to say about this whole thing. The combination showed me that I was not going to be happy long term in any field where empirical evidence wasn't a thing and where I'd be inside all year. So despite the long story short, a bit of a science teaching mishap in high school that had me convinced I couldn't possibly make it as a scientist, I decided to give it another go. And, well, went pretty head over heels for the field. While I was in undergrad, I got a job at the herbarium, the research collection of pressed dried plant specimens. Herbaria are terribly important in botany, and we can get into that later if someone's interested. Otherwise, I will probably talk your ears off about it. I tend to do that. I got a job at the herbarium there, and after I graduated, that led into a job digitizing a herbarium out in Idaho. Holy crap, if I thought I'd fallen head over heels for botany in Ohio. Idaho is, well, the whole Western US has enormous amounts of botanical diversity that are just undescribed. Whatever indigenous knowledge there was of them has been wiped out, genocide and all. And European science has been there for 100 years, maybe 150, and never in very high population densities. My 3rd year in Idaho, I went out with a colleague to check out this plant that this colleague had recently discovered. It's 18 inches tall, is conspicuously different from its closest relatives, grows 5 minutes walk from a major highway in an area with cattle troughs everywhere, so not exactly unknown to people entirely, and was brand new to science in 2011 because nobody who knew how to spot it had ever looked. Idaho botany is just like that. It's wonders everywhere. I could still walk into the College of Idaho herbarium and point out 5 or 6 folders of specimens that we're pretty sure this needs a name. It's an unnamed species, but nobody's had time to look yet. Also, while I was out there, I got to know Barbara Erter. Turned out to be a very good close friend, and also she literally wrote the book on these plants, the For North America section on Potentilla, the genus. And she started suggesting that maybe if I was interested in going back to grad school, I could maybe study these guys. There's a lot to be done in Potentilla. And I was a little skeptical. I thought a lot of the things that we needed to do in there um, would require a way to reconstruct lines of evolutionary descent in plants that hybridize like crazy, which as of 2015, 2014, when I was applying to grad schools, as far as I knew, that was not a thing that could be done. And then I visited UW-Madison, and I met Cecile Annay and Brett Largett, two professors who work in the botany department, a great big chunk of whose CVs, whose whole academic opus is centered on figuring out how to do exactly that. So, well, what was I going to do? I'd already fallen for these plants for the landscape they live in. I knew I'd have some serious experts at my back trying to figure out what the hell is going on here. And I had learned that the tools actually did exist. So off I went.
So let's talk about these plants. You've indicated here that they are pretty special, even in the extremely diverse plant world. I want to give a close-up. Is there a particular image that I could do for this while you describe what's so awesome about these plants?
The brewer's cinquefoil close-up image I sent you, that would be a good one.
Close-up brewers?
Yeah, that's the one.
All right, awesome. I've got that open. So
That's a pretty close shot of brewer's cinquefoil, one of the 5 core species of the group, in a little glacial cirque up in an Uskai Island in Nevada.
Okay, so can you tell me like some of what's going on here with the plant? Because I don't know what to look for. Right?
That's fair. That's fair. That photo is really just a shot of the leaves.
Mm-hmm.
Note how deeply divided they are, kind of feathery looking,
Mm-hmm.
and how silver. The silver color comes from real fine hairs on the leaf surface. The fact of feathery leaves and silvery hairs in themselves isn't all that special. You find those characteristics in a bunch of other plants.
Mm-hmm.
It's which plants we find those particular characteristics on within this group, and where they are and how they relate to each other genetically and from what we can reconstruct from where and how we find them, that suggests we've got, like I said, a group where the big forces of plant evolution are being deeply weird. These plants have next to no immediate economic significance. If you believe that everything was put on Earth for human benefit, honestly, the primary benefit of these plants is to remind us that not everything is, in fact, about us. They're closely related to strawberries. They're closely related to shrubby cinquefoil, which is in the horticultural trade. Very pretty thing. Deer resistant too. But the primary value of my research is the pure research side of figuring out how any of this works and how we can expect it to work in the future as the massive ecological changes we're currently causing really start to sink in. I'm reminded of a friend of mine in the department, another grad student, who studies these beautiful little bulb flowers in South Africa. Dozens of species within an area of like 10, 20 square miles. Some of them only in very small individual spots. Like you can find an entire species that doesn't get outside a 20-meter radius. And their climate niches are similarly narrow. They have to have exactly the right combination of rainfall and temperature, and when the temperature is high or low at which times of year. With current climate change, those climate niches are moving rapidly. If they move so much as 50 miles in 100 years, these plants' only means of seed dispersal is by just dropping them. Mm-hmm. The plant grows a stem that maybe leans a little bit, so it's directly below the flower is 6 inches from the base of the plant, and the seeds develop and fall. So this plant can move maybe 6 inches in a year. Given the current modeled extent of global warming, these species are toast. The hope of my research, the hope of a lot of research in plant systematics, is that we're all piling up straws, a little bit of knowledge here, a little bit of information there, and eventually, hopefully, we can build up, to stretch the metaphor, enough of a haystack that we can climb up to the barn loft of some idea how to maintain biodiversity through what's coming.
Wow. Yeah, that's a lot.
It is a little depressing.
Yeah.
It's been said that to be a naturalist in the 21st century is to live in grief, to watch the critters we love systematically destroyed. That said, that's not what I'm thinking about all day, every day at work. Otherwise, I would not have lasted in this position at all. It's also just, these are fascinating puzzles. The DNA data that I get from sequencing, well, from extracting DNA from one of these little leaves and sequencing it, suffice to say, I spend a lot of time at work swearing at computers. The data itself is tricky, and most of the software for handling DNA data is either extremely expensive or was developed by scientists, not computer scientists, on grant funding that didn't really provide enough time and funds for good beta testing. So just getting the software to work can be a royal pain in the butt. Much less figuring out how to use it and how to get it to continue working with a really unusual data set.
So I know in our previous conversations, you've called the species that you are studying model organisms.
Yes.
Since I come from more humanities and social science backgrounds, could you explain what a model organism is and why that's important?
The prototypical model organisms are things like mice or E. coli bacteria. A model organism is any organism that has a particular trait that you're trying to study. The ideal model organism for studying a particular trait is easy to take care of, easy to maintain in the lab to keep alive and healthy, easy to find, and long story short, easy to research and easy to study these specific traits. These are not prototypical model organisms for, say, plant physiology, because, well, their genomes are completely ridiculous. They have unreasonable numbers of chromosomes. But they're great for studying these big forces that I've mentioned, hybrid evolution and Sky Island biogeography, the particular types of trait evolution that get us these weird leaves. They're great for studying all those things because they are pretty easy to maintain in the lab. The greenhouse has not really had any trouble keeping my plants alive, nor have I, which is saying something. Where was I going with that? Sorry.
Model organisms.
Yes. So these are model organisms for that whole intricate, interconnected play of evolutionary forces that isn't really found in a whole lot of other places. And some of those places are things like, say, giant sequoias. Good luck raising a giant sequoia to maturity in a human lifetime in a greenhouse. Not going to happen. These guys flower in, say, 3 or 4 years, and, well, they don't get a whole lot bigger than this. The flower stems get a good deal taller. This guy is just finishing blooming, but they don't get much bigger.
Awesome.
Actually, if you could pull up the photo of the brewer cinquefoil in its in habitat, that's about as big as they get.
I need to find it. In situ. Luckily, I know what that means. There we go.
Sorry about that.
No
At least
worries.
people have photos with Latin names.
True. For those who maybe aren't super familiar, Alexa and I are friends. We have gone on walks together, and something that is common in our walks is Alexa stopping and going, ooh, and from my point of view, I'm like, is it a bunny? Is it some kind of animal? Is it something cute? And she will spout out the scientific name of the plant and tell me that it's like shoots are starting to come through. She'll give a lot of information that I absolutely cannot repeat without butchering in my attempts, and she'll just laugh at me probably.
No.
So I had a trivia game one night as well, where one of the questions was about what plant blah 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 comes from, and she was able to name like the genus off the top of her head. So
the species, but close enough.
Yeah. So let's look at this picture of the brewers in situ.
So that clump of plant is maybe 2-3 feet across. That's as big as they get. That's as big as I've seen them. That's actually an extremely happy plant, growing in a little valley where there were more individuals of this complex than I've ever seen in one place in the wild. It was pretty great.
So
incidentally, another reason. Sorry, go ahead.
No, you.
Incidentally, another reason I like studying these plants is that they have wonderful taste and habitat. They are restricted to very sunny habitats and seasonally wet habitats above a certain elevation in the Mountain West. So basically, seasonally wet meadows, which are freaking gorgeous.
Mm-hmm.
Eileen, if you could pull up some of the habitat photos, the Big Meadows one, maybe the Jewett Lake one.
All right. Let me pull
These
this
are
up.
these are the places I get to visit a couple times a year. Well, more like a couple weeks in summer. They have very short growing seasons.
I pulled up this beautiful meadow picture.
Yeah.
That's gorgeous. Let's see.
It's a real privilege to be able to go to places like this and not only enjoy them, but do something of value there.
And here's the Sky Lakes as well. Wow.
Yeah.
Something that I did notice from the previous picture of brewers in situ, pulling that back up, there are a lot of rocks around. So you talk about how they're found in meadows, but obviously if we looked at that map earlier, basically they're found in mountains, right? You've gone to the Sierra Nevadas, you've gone to Oregon to collect these plants. Mm-hmm. So I'm interested in like the boulders that we're seeing here, what kind of soil these plants grow in.
It tends to be very shallow, often rocky, sometimes outright gravelly, but with a lot of organic matter between the rocks. So rich soil, but not a whole lot of it, if that makes sense. A thin layer of the really good stuff over just solid rock. Let's see, what was that one called? The close-up of the Steens, the Habitat Rock Crevice. If you pull that up, sometimes they literally just grow out of cracks in the rock. Those are seasonally wet because as the snow melts higher up, it flows down through the rock cracks and keeps things nice and moist. And if the crack's deep enough, a fair bit of soil can actually accumulate in those things. But it's literally just cracks in solid basalt bedrock. and they poke out of it and bloom very happily. They're tough little critters, delicate in unexpected ways, but the habitats that they favor, I have come to many of them in July and found snowbanks. It's cold up there.
Wow.
And snowy.
Yeah.
They don't bat an eyelash at Wisconsin winters, that's for sure. As long as you can keep enough snow on top of them. The problem we have here is sudden thaws.
Mm-hmm. And then refreezes.
Yeah. They can deal with, 20 below as long as there's 6 feet of snow on top of them, because snow is actually an excellent insulator. It may be 20 below on top, but it's not going to be that cold at the bottom of the snow pile.
And speaking about the weather conditions for them, we do have a few questions in chat if you're willing to get into those now. The one that directly relates to that is from Papaya Pop, and that is, what altitude range are your plants found?
It gets slightly higher elevation as you get farther from the coast, but I've never found them below about 6,500 feet. Okay. And the highest, well, I wouldn't be at all surprised if you could find a meadow at these elevations, if they would go well above 12,000 feet.
Wow.
I've collected them at about 11.
Wow. Yeah,
that was an interesting hike. We did a lot of panting.
So
that said, botanists on a hike, we never run out of breath. Yeah. We just spot something fascinating by the trail that absolutely must be examined right
now. So that is also an interesting point that you've talked about. You have had to come up with new mathematical concepts, had to do work with programs and DNA sequencing, but you are also getting out there in the field doing this research.
Yes. I tell people that I spend my winters mostly on pipetting tiny amounts of liquid from one test tube to another and yelling at computers, but that's an oversimplification. What I do in the lab is on some level very interesting, but what really makes it worth it is summer, field season. I get to usually rent a vehicle because my little compact car would not do terribly well on some of these forest roads in the middle of nowhere that have ruts yay deep. I rent a vehicle and go hunt down my plants for a couple of weeks, hopping from Sky Island to Sky Island, hiking, sometimes backpacking. It's really quite nice to get to do that. Not just because I like backpacking, but because the farther you get from a road, the less likely anyone has collected there before, has observed those plants in scientific fashion.
So I promise I'll get to the 2 questions that we had before, but Papaya Pop has asked another question that directly relates to what we've just talked about, and his interest is in which mountains you are finding these plants. So we have said the Western mountains, we've talked about the Sierras. He's wondering if it's also the Sierras and the Cascades, if you get some in the Rockies. Which mountain ranges are you finding them?
This group doesn't get as far east as the southern Rocky Mountains, Colorado, and farther south. They do get into the northern Rockies in Canada, but mostly I'm finding them in the Sierra Nevada, the Cascades, especially the southern Cascades, the Klamath and Siskiyou Mountains in northern California, even a couple spots in the California Coast Ranges, and then in these little isolated scattered mountain ranges, Gearhart Mountain, the Ruby Mountains, the Warner Mountains, the Wallowa Mountains, the Strawberry Mountains, these smaller mountain ranges scattered across the Great Basin, which is, if you haven't heard of the Great Basin, the continental divide in North America actually splits. And there's a big chunk of Idaho, Nevada, Utah, I think it gets into New Mexico and Arizona, small parts of California, that never flows to the sea. Some of it dumps into the Great Salt Lake. Some of it just goes to salt pans where it evaporates into the air. But these rivers don't actually connect to anything that goes to the sea. Scattered across this desert, which is, a lot of it is the iconic desert of the American West. The Area 51 is in the Great Basin. These great stretches of sagebrush barrens, which are really quite lively and biodiverse in their own right, but you have to know where to look and how.
Mm-hmm.
But in the mountains scattered across this place is where these guys grow.
Okay. So I want to get to these earlier questions. We've got more questions coming in, which is awesome. I love this.
Oh,
yeah. When you were talking about working in the herbarium before, we got a question of what is an herbarium? Is it just an arium for herbs? What goes on there?
Well,
If you can go into some details there.
it's just herb as in plant. That's basically what that means, a non-woody plant. But around, I want to say 1300, 1400, someone discovered that if you press a plant flat between two sheets of paper and make sure that it dries out all the way, and you glue it down to a sheet of good quality paper, the standard size is about 11 by 17, and then you store it in a dry place away from all insects, they last a really long time. We literally have specimens from the first herbarium ever created, 700-800 years old, and they're perfectly usable. Color's the first thing to go, but all the small details of structure are still there. You can do a great deal of taxonomic work from the specimens in herbarium. As we get better and better at extracting DNA, you can actually get, at the moment, you can reliably get DNA from a specimen 50 years old. We're getting spotty DNA results from 100, 150 years old. And as we get better at making sense of the fragment, DNA breaks down over time. So as we get better at making sense of fragmentary sequences, that is only going to extend. Not only that, a herbarium never, only the smallest herbaria settle for a single specimen per species. The ones I worked in had anywhere from like 3 to, God, for the really common species, we'd have 50 sheets of a given plant, because each one records the plant in a place at a time.
Mm-hmm.
By going into a big herbarium, the one at Paris has well over 1 million specimens, and looking at which sheets have flowers on them and what date they were collected, you can actually watch flowering times change as, say, the earthworms currently, or you can track the Little Ice Age. You can also reconstruct the habitat that was, say, where a city center is now. If someone collected a bunch of plants from there, you can see what was growing in what is now downtown Madison.
Wow.
They're very important for all kinds of botanical and ecological research.
So, since I have more of a background in anthropology, one of the things that I'm equating this to is that it's just as important with plants to record all of that in situ information, and not just, this is a member of the species, cool, but actually, here's where it was, here's the time that it was collected, all of that information, as it is for indigenous remains and artifacts.
Upside down. This is my field book. Ironically, I coughed up for the waterproof paper, despite working in an area where I've been rained on all of twice. But here's an example of the kinds of notes I'll take for an individual specimen. Not only a great deal of detail on where I am and how I got there, but latitude and longitude to 5 decimal places.
Wow.
That gets me to the spot that I collected this plant within a few meters. Quite frankly, if civilian GPS would go more precise than that, I'd do it.
So moving on to a new question here, which is actually one of the first questions that were asked. But Simon Ashter had this really interesting question about your friend's research, that they only are in a very small area, right? So their question is, if there are whole species that don't exist outside of a 20-meter radius, how does one not get overwhelmed at the amount of species possibilities?
A lot of the time, you frankly do. South Africa is something of a special case. Plant ecologists, specifically a guy named Armand Takhtajan, divided the whole Earth into forestic provinces. A forestic province very broadly describes the habitat types and the ecosystems that you get among plants in an area. For example, there's a forestic province for pretty much the entire eastern half of North America. There is an entire forestic province that fits in not even all of South Africa. It is an incredibly diverse place. It's calling it a biodiversity hotspot is a bit of an understatement.
Wow.
But even in the Western U.S., no one person could deal with it. No one person could possibly wrap their head around all the diversity there is out there, much less describe it with any kind of rigor. So it's a damn good thing there are a lot of scientists.
Mm-hmm.
By specializing by maintaining a certain degree of broad scale knowledge, being able to recognize other plants to genus, even if I can't recognize individual species, by having a rough idea what grows in which habitat niches and how they interact, I can keep a good sense of where the plants I specialize on fit in. But then I specialize on a small group and try to make sense of it in the kind of detail that would be impossible if I was looking at, say, everything in the rose family. These guys are in the rose family. Which also contains things like apples, peaches, cherries, plums, about half the fruit you find in the grocery store, strawberries, raspberries, blackberries.
So in short, wait, half the fruit in the grocery store is rose, basically.
Yeah, pretty much.
So we're just eating a whole lot of different roses.
Rose family.
Yeah.
The same way that the same way that cats are in the same family as leopards.
Okay. I need to wrap my head around that. And while I do that, I have a feeling I know your answer for this, but Simon Ashtier did ask, do botanists and mycologists get along and compare similarities of their fields?
Oh God, yes. It used to be that my botany and mycology the mycology was just considered a subset of botany. But fungi are very different from plants. The DNA evidence has more recently shown us that fungi are actually closer related to animals than to any plant. But plants and fungi share a lot of niches. The prototypical example is mycorrhizae, which are the most important organisms nobody's ever heard of.
Yeah, I have no idea what that is.
Yeah, I was about to get into that, sorry. Plant roots underground, quite a lot of plant species, most of them, I dare say, have symbiotic relationships with soil fungi. The fungus actually grows into the root in a lot of cases. The plant feeds the fungus with the sugars it makes from photosynthesis, and the fungus helps the plant take up soil nutrients. There's actually some evidence for a degree of communication underground. Plants will trade nutrients and sometimes even hormonal signals through their mycorrhizae. Of all things, James Cameron's Avatar did a halfway decent job of a sci-fi portrayal of mycorrhizae, insofar as any of what was portrayed there was possible. The fact that they depicted it as happening between plant roots was refreshing. The mycorrhizal network of any given forest is incredibly important. Our forests would not look anything like they currently do without mycorrhizae. Hence, most important organism nobody's ever heard of.
Wow. And I really like Simon's last comment here. They were reacting to the rose family and just how much we depend on them for fruit, basically. And so their comment was, I was just wondering the other day why we don't see a lot of rose flavored things in the grocery store. I guess we do.
We kind of do, yeah.
But they did
Also,
have another question.
We're not Persian. You head into a Persian grocery store and rose water becomes a lot more important. Go on.
So Simon also wanted to know if you could tell at a glance the difference between plant DNA and animal DNA the same way you can tell at a cellular level.
No. Just looking at 2 vials of DNA, completely indistinguishable. They're both clear liquid.
And then when you sequence it, though, is it obvious at that point?
Some sections of the genome, yes. Others, no. Others, the mitochondrial DNA, for example, the DNA of the powerhouse of the cell, is different, but subtly. Chloroplast DNA, the DNA associated with the organelles that do photosynthesis, is obviously very different because animals just don't have it. And any DNA sequence from a plant versus an animal is going to be distinguishable, but not necessarily at a glance. There's also the problem of what does at a glance mean when you're looking at a giant string of A's, T's, C's, and G's.
Yeah, true.
But they are distinguishable, but it takes a little more work.
Okay. So I kind of want to get back then into the physical going out into the field thing, which aligns great with these 2 questions from Papaya Pop, who I know also loves being out in nature. So he says, how do you find the locations for your plants? Do people just go on random walks?
Herbaria. Some of it is, well, if I'm looking for a particular species, I go to a herbarium. Ideally, I go to one of the databases that people have been building for the last 10, 20 years of herbarium. A herbarium database, you type in all the data from each specimen, and ideally then you also take a picture of the specimen and put it up online, and suddenly your herbarium is searchable. It's enormously powerful. Expensive to actually pull off, though. Typing all those labels is very labor intensive. Building a database like this is what I did for a job in Idaho. So I will search one or more of those databases for a specimen of, say, the Steens Mountain cinquefoil that someone's collected before. And if it's a recent and reasonably a reasonably well documented one, which becomes more likely as they get more recent, it'll tell me exactly where I can go to find it. Probably, because plants individually don't move, but if the specimen's from 1970, the population may very well have shifted a few feet or a few miles. Not usually a few miles, but they don't always turn up where you expect them. As for people doing random walks, yeah, actually, there's a lot of that. Botanists will just go out to explore, and if they find something odd, they will collect and document it. Or if they're in an area where nobody's collected and documented anything, they might do what's jokingly referred to as hay baling, where they collect one of everything. Just the other day, actually, last weekend.
I was just gonna do a follow-up question and ask you about this.
Over Memorial Day weekend, a friend of mine and I went out hiking in northern Wisconsin, looking for some place deserted, where we do in fact have a grain of sense where not spreading virus is concerned. And along the side of the trail, I spotted a field horsetail, just growing, doing its thing. The thing about field horsetails is, oh hey, I've actually got a sketch of one. About field horsetails is they have separate fertile stems and sterile stems. The fertile stems have sporing cones on them. The sterile stems don't, and only the sterile stems are green. They do all their photosynthesis with the parts that don't reproduce. I found a population of field horsetail where every fertile stem had a little ring of green side stems at each joint. This is not a whole lot less weird than finding a population of humans with fully functional wrists and hands growing out of their crotches. Needless to say, I documented that.
Mm-hmm.
Unfortunately, it was on power company land, and was quite frankly going to get mowed into the dirt the next time the company needed to needed to do much maintenance. So
Mm-hmm.
we didn't have much to worry about in terms of damaging the ecosystem. But I'll get back to that in a minute, actually. But yeah, I collected some of those, and currently trying to figure out how to get a plant press from the botany building with the whole thing shut down.
Yeah. So basically, a short version of this is to have the background knowledge so that when you are out in nature, you are able to identify, recognize when something is odd or something is unfamiliar.
Yeah.
Cool. And you've mentioned it a few times, and so I feel like we shouldn't ignore the elephant in the room. It's also basically the whole reason why I started this series. And you're a grad student. I kind of want to ask, if you're willing to answer, about how the COVID pandemic has affected you.
I'm pretty lucky in that I am currently on a research fellowship from the National Science Foundation, of all things, and my funding is secure through September. So I don't have big financial problems to worry about. Don't really have financial problems to worry about. That said, it's very difficult to do any herbarium work when the herbarium is shut down.
Mm-hmm.
It's difficult to do very much greenhouse work, though fortunately I've been able to work with the greenhouse staff, who are, I reiterate, pretty awesome, to go in there. And you probably saw these little mesh bags on the plant.
Yeah.
These are because something like 30% of the plants I have in the greenhouse are all blooming at once right now. So I'm doing some hand crossing experiments, dabbing pollen from flower to flower and seeing how hybrids with known parents come out.
How often does that happen?
Well, how often does it happen that they're all blooming at once?
Mm-hmm.
First time.
Wow.
And of course they opted to do it during a global pandemic. I've had some of these things in the greenhouse for 4 years.
Yeah.
Irony. I definitely can't get any lab work done right now. No DNA extractions, no sending things off for sequencing.
Mm-hmm.
I can do a lot of my analysis work because I can access the lab computers remotely. I've got a lot of the software and the data on my own computer, mm-hmm. and the computing cluster on campus is also available remotely.
Okay.
So I can get some of my work done, but even all of that is strictly winter work.
Mm-hmm.
I don't dare go into the field this year.
Yeah.
Even if I could keep myself safe, I would be an element of the big city petri dish, traveling to these towns. Half the places I stop to get gas when I'm going between field sites have population 20. Mm-hmm. Counties where they have a total of 6 ER beds. I refuse to be the one who introduces COVID-19 into those situations.
And that's something that you don't think about, right? Where, like, you work with plants. You should be safer and, like, all of that jazz. And when you're out in these meadows, you're typically alone, right? So in case...
Hey, if I can get out into the meadows without contacting anybody, there is no better social distancing than being the only human in 10 miles.
But the problem is the getting there, right?
Exactly.
That there are these additional secondary variables that affect both your research and also your chance of being a vector of infection.
Yeah.
And you were... background knowledge, I know a lot about Alexa, but you were going to go to Alaska this summer originally, right?
Yeah, I was TAing a class where we would have taken 16 undergrads and grad students up to do some botanical collecting and some exploration in Alaska, exploring the flora of Beringia, which is the Bering Land Bridge that during the last ice age connected Alaska and Siberia. It was going to be thoroughly awesome. We had some great plans. We were going to visit some amazing places, collect some very real data. We were going to, assuming we, didn't get pandemiced out or something, the plan was to publish a paper with all the undergrads, with everyone involved. But even if you discount the petri dish that is flying 16 people to the other end of the continent, we were planning to spend a good week in Nome, which is the kind of town that literally has 3 hotels total. And which you have to fly to Nome. There are no roads. Okay. There are roads out of Nome, but they don't connect to anything that goes outside Alaska, or even as far as Anchorage.
Wow.
Again, we couldn't in good conscience do that.
Yeah.
And then, of course, the university officially shut down, and we weren't going to be able to get the funding, even if we had decided we could in good conscience do that. But
yeah. And again,
we were also, I was also actually planning to go potentail hunting in Russia, not for my study plants, but for closer related ones in the Altai Mountains, which are weirdly ecologically close to the Central Rockies. That was also going to be pretty wonderful. Build some collaborations with Russian botanists, get acquainted with an entirely new and yet strangely familiar ecosystem. But again, no way.
Yeah.
Not this year.
And I do want to give the caveat that both Alexa and myself agree the right thing to do was for the university to shut down and do whatever they could to increase the likelihood of people isolating, staying apart, and reducing chances of infection. But I think it is important that we talk about things like research and how they've been affected as well. So
I regret that the university had to shut down.
Yeah.
I don't regret that they did, just that they had to.
Yeah. I really like this question, and I think you're probably wanting to get out your field notes again. Simon wants to know, you said the data from each specimen when you were talking about your field notes. What are the categories in, like, the search? What do you write down in your notes? Root, stem, leaf, soil, bloom? Like, what all do you put in there?
I put down precise location and several categories of not so precise location. Country, state, county, down to, in some cases, directions. You need to know exactly how to find this spot. Just writing the coordinates, technically enough, but I mean, if I tell you to go to 43.234 north and 116.585 west, you have no idea where I'm talking about. So human readable detail.
Yeah.
And then information on what kind of habitat it was found in. Let's see. For example, flat, seasonally snowmelt wet meadow at the bottom of a cirque or small hanging valley. Dense vegetation cover, but plants are very short, not counting emergent escapes, mostly less than 2 feet. With common names. Yarrow, timothy, a small, what's a common name for astragalus? Local weed. A small local weed dominant, also some Indian paintbrush, fescue, a small lilac colored thing that looks like an aster, but those are really hard to cite. Things, where I found it, what the conditions were like, and what else was growing there are the big categories. I'll also record any details about the plant itself that aren't going to survive being pressed. The color of the flowers, maybe, or how big the whole plant is. If the whole plant is too big to fit on a sheet, and I'm just going to clip part of it off, which is ideal anyway, because the primary rule in collecting is that you never take more than 1 in 20, 5% of a population. Ideally, you take a lot less than that, but 1 in 20 plants or 1 in 20 stems, 1 in 20 flowers. That's a good rule of thumb for making sure that science isn't going to wipe out the plants that you're studying. It has happened. Not recently, fortunately, as far as I know, but it has happened. So that was a bit of an aside, but you record anything that isn't going to press.
Okay. But I think that's an important aside, right? That as much as we want to study and understand these things, understand their impact, importance, whatever the case may be, to the world or to our knowledge of evolution, whatever it might be, that we shouldn't be destroying what we're studying in the process, right?
Absolutely. That's terribly important, especially when you're studying rare plants like these. One of the little guys I brought in today is, I think this is probably a new species. It's found only in the Wallowa Mountains of northeastern Oregon, and if I had to guess based on maps of the area and satellite photos, I would not bank on it having more than 50 total square miles of habitat. That's probably a conservative estimate. I have collected a few of these, but again, never in places where I was taking more than 1 in 20, because there's a very real possibility that trying to study this too intensively could kill the entire species.
Yeah.
Before it even has a name.
Wow.
I refuse to be the one who does that.
Yeah. So I want to give chat some time to ask questions. But in the meantime, since I do think people in chat, we've got Josie, you hasn't asked a question yet, but they have expressed that they're a plant fan. Also, hi from the Netherlands. Awesome that you're here. And they mentioned that they eat a reasonable amount of rose hips in the Netherlands.
That's nice.
Yeah.
You can't buy those, but they look delicious.
Yeah. In jam and syrup form, apparently. Yeah. But because we do seem to have quite a few people who are interested in plants, interested in even just like going out in the field, learning more information, I would like to ask you if you have any advice for people who want to get out in nature, especially like right now.
Go hiking. Find the wildest place in your immediate surroundings and go check it out. Ideally, do it several times. Hike the same trail weeks or months apart. Watch how the plants change over time. There are quite a few plants I can actually ID in the dead of winter. I wouldn't have had a snowball's chance of recognizing them if I hadn't seen elsewhere the same individual in summer and winter. If I can ID it in summer and then I see it in winter, I can learn what the winter characteristics are. But go hiking. Stay on the trail, please. Since outdoor activities are so much less risky than indoor activities right now, I worry about people widening trails and picking wildflowers when they don't know what they're doing.
Or potentially stomping and killing plants.
Oh, don't get me started.
By going off the trail, right?
Yeah. Trails are sacrifice zones. We select a narrow strip of habitat that is going to be obliterated for the sake of letting people encounter, appreciate, and learn to appreciate and love the surrounding habitat. Just keep the sacrifice zone narrow, please. But do go outdoors. For God's sake, go outdoors. It's beautiful.
Yeah.
There is a surprising amount of diversity even in a city park. Lawns themselves, unless somebody's been really, really careful and put a lot of work into it, are almost never just grass. There'll be little veronica, little chickweeds, little violets and dandelions, and surprising amounts of diversity in your own front yard. If you know a plant fan, try going out hiking with them. Socially distanced right now, wear masks, But I've found no better way to learn to identify and recognize the plants of an area than to go out hiking with an enthusiast and to get yourself a good field guide and just, if you spot something you don't recognize, look it up.
Mm-hmm.
I can list some good field guides for Wisconsin, for Ohio, and some for Idaho. Unfortunately, most of the really good resources I know for areas other than that are more technical. They're floras, which list every plant that grows in an area and requires some botanical background knowledge to learn how to use. But if anyone is interested, I could give you some of those.
I love that you mentioned like looking into a lawn, because right before you did, Josie put in chat, is it silly of me that I'm taking notes to try and, in a minimally scientific way obviously, use on the plants and weeds in my garden? Especially the weeds seem to change per year. For example, last year was a big redshank year, and this year they're not here at all.
Cool.
So,
That's
like,
no, that's
let's
that's
answer
not,
this question. Is it silly of Josie?
not remotely. God, no. I actually have a little green notebook where I mentioned when spring comes late, I'm jonesing for green. Here in Wisconsin, spring comes about a month later than I'm used to in either Ohio or Idaho. So my second year here, I got myself a little notebook and started writing down the first time I spotted any given species as a shoot or blooming or in fruit or whatever each spring. Mm-hmm. And that way I can flip back a few pages and go, okay, it's only been 3 days since the daffodils bloomed. You cannot expect spring to be in full swing yet. Take a chill pill. But that's also data. Like I said, well, the famous example of the value of that sort of thing is Thoreau's notes from Walden Pond. Give us a remarkably good record of what was blooming when 100 years ago. So taking notes on what's grown in your lawn is not remotely silly. You're gathering very real data, and even if it doesn't end up going anywhere, you're learning for yourself, and that's important. Mm-hmm. I mentioned at the very beginning that a lot of people just see plants as scenery. That's a very real problem, because I can never remember who said this, and I really need to look it up because proper credit. We preserve only what we love. We love only what we understand, and we understand only what we're taught, whether someone else teaches it or we teach ourselves. The only way we're going to reach the future with biodiversity intact is by recognizing it and by coming to recognize, understand, and love it.
Josie put in chat that they have a gardening notebook, but they didn't think to document the weed, since most are welcome to them, growth per year or season. And this is not coming from someone who understands or studies plants, but I've always been of the opinion that weed is just what we call something we don't want to be there.
That is the definition of a weed, a plant where it's not wanted.
Yeah. So there's nothing that's inherently a weed.
Yeah. There are plants that are ecologically noxious weeds. If you've ever heard of Japanese knotweed, in Japan, perfectly harmless native plant. In England, they treat it like literal radioactive hazardous waste because it spreads so ferociously, it chokes out anything else nearby. It is a real menace. Around here, the problems are, the biggest problems are bush honeysuckle and garlic mustard, mm-hmm. both harmless in Europe, but here they can reduce a thriving forest understory to just solid of a single species.
And going back to your advice for going out into nature, when Alexa for the chat said about going out social distancing with an enthusiast, I have gone on walks, safe, during this time with Alexa, and because of that, I can recognize by sight at least one of those species that she just mentioned. I probably can't recite the name
Yeah.
regularly, but I can be like, that's an invasive species, and I know that. And it's only because I've gone on walks with Alexa. And then connecting to what you were just saying, that it's not that a weed is a problem, really. It's if it's an invasive non-indigenous species that it can be a problem.
Yep. And in gardening contexts, around here, stinging nettle is a perfectly harmless native. They actually make remarkably tasty cooked greens, and I'll get back to that in a minute. Don't you dare eat anything unless you know what you're doing.
Also tea.
Tea and cooked greens, yeah, nettles are great. But if you don't want your tomato bed to literally sting you, they're a weed in your garden.
Mm-hmm.
They are kind of unpleasant to grab with your bare hands. I don't recommend it.
And yeah, since you brought that up, let's talk about foraging.
Foraging is enormously rewarding and a great deal of fun and can very easily literally get you killed. You have to know what you're doing.
Mm-hmm.
The best way, of course, is to learn from someone who's been foraging for a while. Learn from them what they know and what they consider to be the boundaries of their knowledge. Take me, for example. I I actually used to scare the crap out of my camp counselors when I was like 13 by picking wood sorrel off the side of trails and nibbling it. Delicious stuff, tastes like lemons, perfectly harmless. But to this day, I refuse to try and hunt wild carrots because wild carrots are literally the ancestors of our domestic carrot, but they look very, very much like water hemlock, a single mouthful of which can kill an adult. One of our most lethal species in eastern North America. So you have to know.
It has killed many people who have done wild foraging or thought that they were collecting carrots.
Several every year, yeah.
Yeah.
Basically, the rule number one of foraging is if you're not sure 100% both of what it is and that species is safe in the way you want to eat it, you do not eat it.
Mm-hmm.
End of story. So that is the thing that you need to know about eating wild. That said, if you say get from a reputable source a young plant of something wild and edible and you plant it in your garden, that's safe.
Mm-hmm.
Just make sure that you know it's the same thing when you go back whenever later to harvest it. There are also some excellent books on wild edibles. I don't recommend that to be your only source to learn how to forage. You should also work with an experienced forager or take a botany class either from a university or from an outdoor education center or whatever opportunity is most accessible where you live. But coupled with a little background knowledge, a good field guide can take you a long way.
Nice. I'm asking for some clarification on a question that was just asked in chat.
Mm-hmm.
You may know what it is, but I don't. They asked, have you ever seen a tree of 40 fruits?
A tree of 40 fruits. I don't know what that means.
I'm thinking that it human engineered plants in the 40 fruit tree. I think it might be okay, yes, there we go.
My best guess would be grafting.
That's exactly it. Have you ever tinkered with species grafting? Thank you, Simon, for posting that right before I put in my question. So it's a human engineered tree with branches grafted from a bunch of different fruit trees that somehow produces them all.
Yep. I haven't done it myself, but I've read about it. And I've seen some, not as many as 40, but I've seen cases where like you have 3 different apple cultivars varieties on the same tree. Grafting is deeply weird and really cool. Basically, if you do it right and you line up the tissue layers of the 2 stems, you can cut a notch in one stem and cut off the tip of another stem, stick them together, and they will grow together. And the stalk, the plant that you attached it to, will keep doing its thing, and the scion, the plant that you attached, will keep doing its thing. You can graft a potato to a tomato and get something whose roots crank out potatoes and whose stems crank out tomatoes.
This sounds like the perfect plant for my diet.
right?
Well, what I want to be my diet, not necessarily
Well,
what it should be.
I'm a total potato fiend. I really ought to try that sometime.
Wow.
Um, you can do it with apples, apples, pears, probably a lot of stone fruits. Those are all closely enough related that you could very easily graft a tree that produces 15 varieties of apple. Um, you wouldn't get very many of each type of apple because apple trees just don't get that big, so you're only going to have 1 or 2 branches per variety.
Mhm.
But it would not be far-fetched at all to have a tree that produces both Granny Smith and Gala, or a tree that produces peaches, pears, plums, and several varieties of each.
Wow.
Yeah, it's cool.
So, if people are titillated by this conversation, I enjoy that word. I blame you for it being currently in my repertoire. Um, do you, like, what advice do you have for someone who is interested in pursuing botany? Whether that be academically or more as a, like, layman.
As I mentioned before, take botany classes, go on walks with enthusiasts, um, read read about it. Good field guides. If anyone has a specific area that they'd like a good field guide for, I can do a little research and see what I can find you. Um, reading books by botanists is also a great way to get some ideas. And of course, now I can't actually think of any. I love that feeling.
Yeah.
Don't think about pink elephants, and all you can think is pink elephants.
In our previous discussions, you've also mentioned that some places have like nature clubs or
Yes.
things of that. Like, what are those? Do you recommend them as things to join?
I absolutely do. The first sort of thing that comes to mind is Friends of insert local park here. The Friends of Hoyt Park, which is a neighborhood park, maybe 3 blocks from my place.
Mm-hmm.
They're busy. They do some remarkable work. They've put up great interpretive signs, those signs that you'll sometimes find in parks that describe what is going on in the nature or the history or whatever of a place. And they've done so much invasive species mitigation. Comparing it to other nearby parks that don't have similarly busy people, Hoyt Park has easily twice the biodiversity. It's pretty great. They take out the bush honeysuckles and the buckthorns and the garlic mustard and let the native plants thrive. Working with a group like that would be a great way to learn a lot of basics. And I mean, not just basics, but certainly a great place to start. Similarly, if you're near a major, like if you happen to be near a national park, I would be extremely surprised if there aren't good community organizations dedicated to helping out with environmental work in the park. If there's a foraging club or just a botany club, if you're near a university, there's probably a student botany club. Speaking of universities, actually, in the US, state universities, I think all of them have agricultural extension services. These are units of the university. They're devoted to helping farmers and other agriculturalists with any questions or problems they might have. They'll deal with, they do a lot of agricultural research. They help describe how to apply it to farming, but they also do things like produce home canning guides to explain exactly what food safety rules you need to follow to not give your family botulism. And many of them have plant identification services. If you've got a plant you don't recognize, they may or may not be accepting mail-ins right now because pandemic, but in the before times, you could just take it to your university's extension service and they would very likely be able to figure out what it is for you.
Mm-hmm.
They would also be able to recommend good books and good resources. If you have a master naturalists organization in your area, or even master gardeners, Those are great places to start. Master gardeners is just as good as master naturalists. They just focus more on horticulture than botany. Look for whoever's doing it, whoever's already doing botany in your area. If you have no other ideas and you're in a college town, or even if you aren't in a college town, email a botany professor, see if they get back to you. If they aren't insanely busy and they do get back to you, the vast majority are going to be super enthusiastic. Certainly I would be.
So I don't want to like inundate you with requests, but I have texted Alexa pictures of plants before, and at the very least she's been able to get me the genus. If someone wished to contact you, again, please don't inundate her with like a ton of pictures of plants. She loves plants, but just like keep it reasonable, I would say.
It's fun stuff.
Yeah.
I actually may or may not hang out on Reddit, what's this plant board, trying to ID photos from all over the world.
Yeah. So where can people
in this hemisphere? But sorry.
Where can people contact you then?
I'm on Twitter. I believe you have my Twitter handle, photosynthetic4. And I have a website, alexadinicola.com. I'm also photosynthetic on Reddit, so can reach me at either of those.
Awesome. And just real briefly, since we mentioned it before about don't eat things unless you're 100% sure you know what they are. Same thing goes for mushrooms, fungus, anything in that family. Just if it's in nature and it is a shroom of some kind, just don't eat it. Don't touch it.
Yeah.
Even professionals who go out foraging for mushrooms sometimes get it wrong and die. So just be very careful on that said. But I like this quote from Simon Ashtier. They're quoting you saying deeply weird and really cool. That's precisely what I'm here for. I think that's exactly what we got. This was awesome. Alexa, hopefully people follow you on Twitter, because there's even more awesome content there. If you have additional questions, you can go ahead and reach out to Alexa there. Check out her website. And yeah, is there anything that you'd like to add, Alexa?
I think you've pretty much covered it. We've pretty much covered it. The questions have been great. You guys have you guys already seem to care, and that's the most important thing. Caring enough to be curious enough to go out and look at what's there.
So
that's science in a nutshell, really.
And you know what? If you do go out into nature, follow any of the advice from Alexa, I think that you'd probably be okay with me saying this. Take a picture of yourself out there, tweet her.
Hell yes.
Yeah.
Absolutely. I'd love to see that.
Yeah. So for those who follow my channel regularly, I'm hoping to turn this into a regular series. I've got a few other people that I've asked. We just got to work out the time. So follow me on Twitter as well at GreenEyeTbone. I'll put that in the chat. And that's the best place to go if you want to find out when our next Twitch stream with an awesome grad student will be. For now, though, I think that we're going to sign off. Have a wonderful day. Stay safe wherever you may be. Reach out to us on the social media. We'd love to hear from you. And we'll see you later. Bye.
Thanks again.